All right. Right now we're going to look at Gregor Mendel. We'll look at some of the patterns of inheritance that he discovered in his experiments. So let's jump right into it. All right, Gregor Who. Look at this handsome devil over here. So this is Gregor Mendel. He was an Austrian monk. Don't mind these numberings over here; they're way off. So he came up with the original theories for heredity and also based upon his experiments. So it took him 7 years to do crosses with pea plants. He did tons of different crosses during that time. As you can see, almost 30,000 pea plants that he tested on between 1856 and 1863. He produced, he crossbreeded honeybees as well, produced a hybrid strain of honeybees that were so vicious they had to be destroyed. So it's kind of cool. So he was a monk, but he was also interested in astronomy, meteorology, and even the study of evolution. And his last words were, "My time will come," and that's because a lot of his theories and his experiments kind of got lost. It wasn't until much later that his initial discoveries were rediscovered, and scientists were able to use them in conjunction with evolution to talk about genetics. So he's kind of the father of genetics. So this is a simplified version of his studies. What he did is he did studies with the pea plants, and he would keep one trait. So let's say he just wanted to test pea color. In this example, it's when he tested the yellow and green peas. And what he'd do is he'd start with parents that had opposite traits. So the mom plant or the dad plant is yellow, and mom or dad is green, and he produces a cross between them. The way he's able to do this is he just takes like he removed all of the male parts from one of the plants, and so that way it couldn't pollinate itself. And then he takes the male parts off the other one, and he dusts them against the other plant. So he takes maybe the yellow male parts, dusts them against the female to get his cross. So that way he's the thing that's pollinating the flowers. And they're not getting pollinated by bees or wind blowing the pollen. He's controlling all the pollination. So that's the first kind of thing that he did, controlled part of his experiment. And then he crosses them, and he looks at the seed color for all of the offspring. So we call that F1. It's first filial generation, but it's basically just the offspring from this initial cross. And you can see in our little branch diagram, they're showing the parents coming together, and then here's all the offspring coming off. Now, his studies were had a background in mathematics, so his knowledge of math helped him in his understanding of the results he was getting. So he crosses a yellow and green plant, and what he sees is that in all the offspring, 100% of the offspring, the green trait disappears, and the yellow trait is there. And at that point, he doesn't stop. He's still thinking to himself, where did the green trait go? Is it totally gone? Why did that trait disappear? So what he does next is pretty cool. He crosses in the same way, where he removes the male parts from the plant. From that offspring, that first offspring generation, he cross-pollinates these plants. And then we get the F2, or second filial generation. So after crossing these yellow plants that all are yellow, what he then gets is a ratio. So he probably did hundreds and hundreds of plants in each generation. You could, if you remember from the last slide, he said he tested on over 30,000 plants, or over 29,000 plants. So what he comes up with, he's got a big plot of plants. What he notices, though, is that 3 out of every 4 are yellow, but 1 out of every 4 is green. So his ratio is 3 yellow plants, 3 yellow plants, to one green one. In Matt's, over all the crosses, he gets this 3 to 1 ratio. And then if you cross the plants out again to a third filial generation, he gets a 3 to 1 ratio of the yellow to green seeds, yellow to green peas. And what that allows him to do is start to make some assumptions about what's going on, especially using your knowledge of meiosis and how those reproductive cells are separating. It allows him to make some, even without being able to know what a gene was, he called them factors. He knew that the factors from one parent are separating and they're being combined with the factors of another parent, and that those factors were then passed on to the offspring, and the offspring would then pass those factors on to its offspring. So let's define some terms and get a little bit better understanding. So we're going to develop a little foundation for genetics. So one of the keywords we use is traits. So traits are the specific characteristics that vary from one individual to another. So you could have a trait for eye color, you could have a trait for hair color or skin color. And then our next idea is, our next keyword is a gene. And a gene is a sequence of DNA that codes for a protein and thus determines a trait. So a gene, if we think back to what we remember from cell division, you have a chromosome. Chromosome is made up of wound up DNA. If you're able to take a portion of that DNA, one section of it, and a piece of the chromosome, you would find a gene. And there's multiple genes on a chromosome. There you go. So hundreds of thousands of genes per chromosome. Then the other important thing that we have is an allele. An allele is one of the alternate forms of a gene that controls a characteristic such as eye color or hair color. So what that means, in Mendel's experiment, what he was really lucky with is that in the pea plant, all of the traits that he studied, there was only 2 alleles. So it could have been a round or a wrinkled pea seed, it could have been green or yellow, tall plant or short plant. There was 2 alleles for each one. And I've shown a couple things. If you look over at the image, and I'd like you to draw this image because I think it's a pretty good one for you to keep. Here's our chromatids, and you could imagine if this was mitosis, there'd be a centromere attached here, pairing these 2 up. So we have a chromosome, or I'm sorry, a chromatid, and on this end of the chromatid here is one allele, and this would be allele for purple flowers. And then on this section, you've got an allele for white flowers. So on this paired chromosome, each chromatid has got a different allele. And this, what we call a locus. Locus is kind of like your heart is found in an area of your body, or the lungs are found in an area of your body or your liver, and in everybody's the same. In human beings, you have a chromosome. The gene for certain traits would be on the same area of a chromosome. It wouldn't be flipped around. So for flowers, this is saying that this flower color gene is located on this specific area of the chromosome. Now, with our alleles, one allele could be dominant and one could be recessive. And that doesn't relate so much to fitness. So it doesn't mean that an organism is going to be surviving better if it has all dominant traits. It only means that the dominant trait will mask the recessive. And the recessive trait will only appear in the phenotype if it inherits 2 of them. So it needs 2 recessive alleles to have the recessive trait. Dominant, you can have 1 dominant and 1 recessive allele, and you'd be dominant, or you could have 2 dominant alleles, and that's shown over here. So we've used B, big B and little b, big B, we always use the big letter to represent the dominant trait, and the little letter to represent the recessive trait. So here we see you could have a big B or little b, or big B, and you have brown eyes. And here you've got blue eyes. Little b is the only way to have that recessive trait. Okay, so look at homozygous and heterozygous. So because the alleles come in pair combinations, we want to come up with a word to describe the different pairs that you could have. So let's say you have 2 copies of the dominant allele, then you would be homozygous dominant. So homozygous meaning you have the same pair of the dominant trait. You could also be homozygous, the same allele, but homozygous recessive. This means you have 2 copies of the recessive allele. And then there's one more type where you could be, it's heterozygous. And if you're heterozygous, you have 1 copy of the dominant and 1 of the recessive. And it doesn't matter which way you write the allele combination. Heterozygous is just showing that you have 1 of each copy of the allele. Now, we're gonna, the genes, although we could look and see, we could see the gene, especially during initial genetic studies, they weren't looking at the genes, they were looking at the outward, what we call phenotype, or physical trait. Now we can sequence genes, and we can look at genes in a chromosome. But the genes in the chromosome are the written information in the DNA. So they don't have a particular color or shape or size. It's only the coding for the information. So for the genotype, it's the particular alleles at the specific loci present in an organism, and the genetic makeup of an organism. So when we looked at that chromosome, there was a specific area for flower color. That locus of the chromosome was where the gene was located on each chromatid. For phenotype, it's when that particular combination of genes comes up, what's it going to look like as the organism is built? So the phenotype is the physical traits or the characteristics of the organism. And they come from the consequence of the genotype. So the instructions in the genes are going to tell, as the cells are being built, what the overall organism is going to look like. So Mendel came up with 2 laws that are still consistent today. Luckily for him, when he tested different alleles and different genes that were located really far away from each other. So if a pea plant was short, that gene was not located really close to the color of the peas, or the height of the plant, tall or short, was not located close to whether it was a wrinkled or smooth pea seed. And this allowed his law of segregation to hold up. And what that means is that for each gene segregating, alleles for each gene segregate during gamete production. So what that means is that in meiosis, the alleles for each gene are going to separate from each other. So our blue beetle has got 2 alleles. When this beetle makes up its reproductive cells, one allele is going to go one way, one allele is going to go other way. So here's the beetles. They made it having a big B, little b. And then we've got our female beetle over here with her eggs, and she's got little b, little b. What it means is that the bees, the different alleles that were present are not going to go together. They're going to go separately, and they're going to donate one possible allele. So blue beetle with 1 each allele and green beetle with 1 each allele, you could put into this Punnett square and look at all the possible outcomes. So if you combine these alleles with these alleles over here, you get 4 possible combinations. And the Punnett square is a nice way to organize all of those possible combinations. The other law that Mendel got was that alleles for one type of gene will segregate independently from alleles of another type. So what that means is that, going back to what I was saying about the wrinkled and smooth pea seeds, is that they're going to separate independent of one another. So wrinkled seeds would go and separate, and green seeds will separate the other way. So they're going to be separating independent of each other. And this is because for the genes that he studied on the chromosomes, these genes were so far away from each other that as crossing over happened during meiosis, they didn't cross over together. They were separate, and so they weren't passed on together, and they weren't linked genes. All right, that's the end for today. Big takeaways, just background on Gregor Mendel, knowing that he's the father of genetics. Really getting comfortable with all the terms. You want to get a really good foundation for those words because we're going to be using them again and applying them almost as if you would apply them in math or geometry. And so you want to be really comfortable with those. And then also knowing Gregor Mendel's 2 laws. And that would be the biggest things to take away from this lesson. All right, see you back in class.